Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientists monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host and moderator for today's webinar. Today, Dr. Marc Caron will present leveraging novel concepts of receptor biology toward a better treatment for schizophrenia. The Brain and Behavior Research Foundation is committed to alleviating the suffering caused by mental illness by awarding grants that will lead to advances and breakthroughs in scientific research. The Foundation is the largest private funder of mental health research grants. Since 1987, the Foundation has awarded more than $346 million. 100% of all donor contributions for research are invested in grants to scientists who are working to find breakthroughs in disorders such as ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, OCD, PTSD, and schizophrenia. I'm delighted to introduce Dr. Marc Caron. Dr. Caron is the James B. Duke Professor of Cell Biology at Duke University Medical Center. He is a recipient of the 2005 Distinguished Investigator Grant, the 2013 Lieber Prize for Outstanding Achievement in Schizophrenia Research, and a member of the Foundation's Scientific Council. Today's webinar will begin with Dr. Caron's presentation. This will be followed by a question and answer period. To submit your questions, please use the Questions tab on the control panel on your screen. Please feel free to submit your questions throughout the presentation. Following the presentation, I'll present your questions to Dr. Caron, and we will address as many as possible in the time allotted. And now, I'm pleased to introduce Dr. Marc Caron. Marc, the floor is yours.
Thank you very much, Jeff, and thank you for all of you that are joining online. I'm delighted to do this, and I would like to thank Dr. Borenstein for asking me to do this. It's really a privilege to be able to do this with you. Let me see, I'm trying to, okay, so I think so as Dr. Borenstein said, I titled my presentation today as Leveraging Novel Concepts of Receptor Biology Toward a Better Treatment for Schizophrenia. So as a point of disclosure, as Dr. Borenstein said, I'm not a clinician. I'm a PhD scientist. I've been interested in G protein-coupled receptors, these receptors that we are going to talk about, for essentially all my career as a scientist, which is almost now close to 45 years or so, almost 50 years pretty soon. And I will try to essentially give you a sort of a story. As you know, it's sometimes difficult to talk science without the words that we commonly use in scientific language, but I will try to mix this with sort of a caption that will be understandable for everybody. If we go to the first slide, I mean, I put in a statement of purpose to basically try to tell you what I think about, I hope I will achieve today. First, I will, if you register for this, you must know that all clinically prescribed antipsychotics act essentially as blocking brain dopamine D2 receptors. These receptors are called G protein-coupled receptors because of the way that they work. We abbreviate them GPCRs. Now, in the story, the thing that I'm going to tell you it did not happen overnight. I mean, this is a story that has evolved over the last, I would say, 20, almost 30 years. But the interesting thing that I would like you to take away from this is what I'm going to use as the platform of my talk today, is that we now understand that depending on the biochemical makeup of cells and neurons in our brain, that GPCRs can engage distinct signaling pathways. That is, more pathways than we had originally thought. And in doing so, they affect many distinct cellular responses, okay? So the major point of my presentation today will be in experiments that we have tried to validate this concepts of receptors working in two different ways in genetically engineered animals. And these studies have revealed that a novel, I call it putative antipsychotic agent, but at this point it's a tool compound, it's a tool agent. But that compound basically blocks D2 dopamine receptors in the motor area of the brain, that is the striatum or the caudate in humans, but activate D2 receptors in the cognitive and executive function areas of the brain, that is the frontal cortex, where most of these functions are carried out. So the bottom line, what I would like for you to take out of this today is that an antipsychotic with these properties might have more effective, might be more effective at alleviating both positive and negative symptoms of schizophrenia. And I'm going to try to develop the idea with my presentation that this may be an interesting thing to pursue. So outline of my presentation, I'll show a few slides basically defining what G protein coupled receptors are. They are essentially the largest gene family in our genome. This is how we see light. So vision is mediated by G protein coupled receptors, in particular called rhodopsin. This is how we smell. That's how we taste many flavors. This is how your heart beats, how your brain cells communicate. Now, GPCRs, as we call them, are virtually expressed in every cell in the body. And I will make the point that they're named because they work through these proteins, so-called G proteins, so they're called G protein coupled receptors. But over the years, we've now found that they also do other things through other components in the cell. And this has led to this property that we call functional selectivity or bias signaling properties of these receptors. And it provides a basis for new physiological principle, but also what I would like to concentrate on today is that they probably give us a handle on new therapeutic developments. So a few slides that I'm going to show you after this introduction is how several years ago now, almost 10 years ago, we elucidated and studied the potential implication of a novel pathway, which we call just by these acronym AKT, GSK3, they're molecules in cells, that is a signaling pathway in the actions of dopamine, which this was not anticipated before. And so we validated this by genetic validation. That is, we did this in animal models, in mice mostly. And then the bulk of my presentation will be how we are trying to leverage this concept of functional selectivity to understand the cell and molecular basis of disease. But I'm not going to concentrate very much on this. But the thing that I'm going to concentrate on is the development of more selective and efficacious therapy. At the end, we have validated also this idea of functional selectivity for the actions of L-DOPA in Parkinson's disease. I'm not going to present this today, but I have some slides if somebody is interested. Now, let's go back to GPCRs, okay, these receptors. So these receptors, as you see here schematically, they exist in the membrane of cells because they are the conduit of information from outside the cell, hormones, neurotransmitters, any kind of activators of these receptors. It leads to a signaling across the plasma membrane because they change the conformation of the receptors that engage these so-called G protein that are inside the cells, and then ensues signals that are, for those of you that know this, calcium, cyclic EMP, and the alike, and you lead to a cellular response. Now, associated with this in just about every cell of our body, when a receptor is activated, there's a mechanism that basically prevents the cell from being constantly activated. And this is usually coined by the term desensitization or tachyphylaxis. And over the years, this goes back to the beginning of the 80s or mid-80s with my colleague, Dr. Lefkowitz here at Duke and ourselves. We basically characterized mechanism by which these receptors, quote, desensitize. And what happened when the receptors are occupied initially to response, with time, there is another set of protein that are called G protein coupled receptor kinases. So these are enzymes that put a little tag on these receptors. It happens to be a phosphate group, okay? And that phosphate group allows another protein, which was originally identified in vision, because that's what I told you, vision is quite important for the paradigm of GPCRs, that arrests this activation, this cellular process, and it blocks essentially the process. Now, one of the better examples that I always tell students in this is that if you go into a bakery, okay, and you open the door, it smells like bread. I mean, it's wonderful. But then you have to wait for 2 or 3 minutes at the counter. What happens is your receptors desensitize and you don't smell that good smell of the bread. You don't realize that you are surrounded by this. So this is the process of desensitization. So that's what these olfactory receptors go through, this very process of desensitizing. Now, what's interesting here is that this was what we were working on 30 years ago. Over the years, we've realized that these receptors, as the name implies, coupled to G protein, these G proteins that are inside the cells and elicit signals because they activate enzymes that make signals inside the cells, or they activate channels that lead to increase in calcium or potassium in and out of the cells. But what we've sort of realized is that these mechanisms that we had basically identified, that is the phosphorylation of receptors by kinase and the binding of arrestin that basically eliminate or limit this signal here, were able to do other things than this. Initially, we showed that this was the signal for the receptors to internalize, to be taken up inside the cells, to remove that little tag here and recycle the receptors as competent receptors. Then a few years later, this goes back to late 90s, we realized that this receptors, which was tagged here by the kinase and bound to arrestin, was capable of initiating signals that was different than this. These signals were usually associated with other types of signaling, like growth factors and things like this. So this was kind of interesting. And in a sense, I mean, we even showed at the time that this signal, the G protein mediated signal, is usually rapid, but this one is usually a little bit slower, but it's much, much more protracted. And so it probably does other things in the cells. And eventually, scientists discovered that you could have agonists, that is, agents that activate receptors or inhibit receptors that will activate or inhibit this form of the receptors, but do the contrary here, or vice versa. So there was this idea of bias signaling or functional selectivity. That is, a receptor can be activated by a hormone, but be blocked. This Barrestin dependent signaling might be blocked by the same hormone. So this led to this concept. So what we now know, thanks to the work of one of our former trainee here, Dr. Brian Kobylka, who has crystallized these receptors in the G proteins, and we now know that they go through the plasma membrane and they have special structure, and the ligand, the activating molecule, usually bind inside the membrane here and activate the G proteins. Now, and send signals that activate a cellular response. Now, what the G protein arrestin interaction does, the other side of the equation, is that one, it desensitizes this signal, but it also activates the pathways different than the G protein receptors. And also, as I just told you a few minutes ago, it internalizes the receptors so that they can be recycled as competent receptors. Now, I just wanted to illustrate for you how interesting what I'm going to tell you about, because I'm going to tell you essentially about one receptors, the D2 dopamine receptors, which is a target of antipsychotic. But just for your interest is that GPCRs are the largest family of genes in our genome. We have about 800 of these receptors. Interestingly, 400 of these receptors are set in our olfactory epithelium for olfaction. Well, as you know, we don't depend very much on olfaction for survival, but other species do. So it means that we now have about 400 other receptors that probably are activated by hormone, neurotransmitters, and many, many different ligands. We call them ligands. But interestingly, there's still about 150 or 200 of these 400 receptors that are still orphaned. That is, we still don't know what and who is activating them. So there's still a lot of work to do, and there's still a lot of potential targets here for various diseases. But the other thing, just to show you that the mechanism that I'm telling you about, the ability of the phosphorylate the receptors and the arrestin to bind to the receptors to change their signaling molecule. Look how this, there are only 7 kinases in our genome. 2 of them are visual, and 5 of them are for the rest of the GPCRs in the periphery and non-visual. And there's also only 4 arrestin. 2 are visual arrestin and are for the rest of them. But then consider that this is an interesting stats for you, is that therapeutic drugs, 40% of these drugs, only or 50 of these receptors. So that means still in 50 receptors of drugs for various conditions. The only problem is that we have to discover what those conditions is. And in many instances, we know what these receptors do, but in a lot of instances, we don't know what they do. And the question here is, the question at hand is, can we leverage some of the recent principles that were identified in GPCR function to improve the specificity and the effectiveness of GPCR therapeutic agents? I'm going to try to develop this for the dopamine D2. Before I do this, I would like to show you something that because I know that everybody knows what morphine is. Morphine is par excellence analgesic or anti-nociceptive agents, and work that Laura Bond did when she was a postdoc in our lab back in the late 1990s, when we characterized a mouse that had one of these arrestin molecules deleted. You can see what we show is that the lack enhances the beneficial effects of morphine, but attenuates its side effect through this opiate receptors. And you can see that this is a response that we see in mice. For example, you put a mouse on a slightly warm plate, and it takes about, a few seconds, like maybe 10, 12, 13 seconds to withdraw its paw. And the effect of morphine basically lasts for almost 2 hours. Well, look at what happened in the absence of this arrestin molecule, is that now the morphine is much better at alleviating the pain sensation in the mouse. So this is kind of interesting, but the thing that's most interesting is that the lack of arrestin alleviates some of the side effects of morphine. And those side effects of morphine are that it decreases respiration rate, and also somebody, as many people have taken morphine for pain management, you know that it causes constipation. So these, when you don't have arrestin in your cells, basically you do not get constipated when you take morphine. This comes to the point that several GPCRs, I would say maybe 10 or 20 GPCRs have been now examined for these principles. But for those GPCRs which we've examined the physiological consequence of this functional signaling, it has been realized invariably the G protein and the arrestin-dependent signaling pathways do different responses to basically are involved in distinct responses. So this is quite and I should say that there's a lot of companies now that are trying to make compounds that are analogous to morphine that will not do or will not induce some of the side effects, but also that will not induce the addiction, because as many of you see or know that we have a lot of problems with morphine addictions, which eventually leads to heroin addiction and things like that. So we hope that this concept, studying this concept for the opiates will eventually lead to drugs that are much better and do not induce addiction. Now, what I'm going to show you here in the next few slides is basically how we elucidated this novel signaling pathway downstream of the D2 receptors and how we use genetic validation to show this. And in a sense, I mean, it's sometimes like detective work, as I mentioned, is that we used animals that we had engineered many years ago. Either the dopamine transporter knockout, and this is the gene, the dopamine transporter is involved in recapturing the dopamine in the brain that is released by various activation of neuron. And these mice are very hyperactive. And we also had generated with the postdoc that was in my colleague's, Dr. Lefkowitz's lab, the Barrestin knockout mice. And we used these mice to question, well, is the D2 receptor, this site of action of antipsychotic, could it be involved in pathways that are using the arrestin and the kinase, and the arrestin is a signaling molecule? And indeed, we were able to provide biochemical evidence and then genetic evidence that there was a pathway here which was different than the ability of the dopamine receptors to inhibit these cyclic EMP and DARP32 that probably exists in the brain, which engages downstream of arrestin a couple of 2 or 3 different kinases that eventually lead to cellular response. Now, I should say that this pathway, not the involvement of the D2 receptors, but this pathway of this AKT and GSK3 kinases in cell had been proposed as potentially being involved in schizophrenia because there were variants, genetic variants of AKT that track with schizophrenia. This pathway, the AKT and the GSK3, are affected by antipsychotic, and we and others had shown that GSK3 genetic overexpression in the brain mimics the schizophrenia phenotype, or if you delete it, it basically recapitulates some of the effects of antipsychotics. And we had shown in cells that the several of the antipsychotics, the clinically effective antipsychotics, seem to be more uniform at inhibiting this pathway. Now, what we have done over the years, and you can see we've published some of these studies in 2012, is use the Barrestin knockout. That is, we use genetic methods, now that I'm not going to go into the detail, to invalidate the gene of the beta-arrestin in the mice. So this is whole body Barrestin knockout. Or we use another trick where we take this enzyme, which has a little tab to delete it specifically in cells. And when we do this, we can show that the deletion of either the arrestin or either the GSK3 in two cells in the brain, in the striatum, in this particular instance, recapitulate the effects of antipsychotic in animal model of psychosis. Now you can say, well, how I know that you're not the clinician, but how do you tell that are psychotic? Well, we sort of borrow some ideas from. So we treat these mice usually with psychostimulants like amphetamine, which we abbreviate PCP, as I will tell you in a minute. And what I point out, and this is also in my next slide, I'll come back to this, is that we're not trying to simulate the symptoms of psychosis. We're trying to make conditions like stimulating with amphetamine or phencyclidine that reproduce some of the things that we presume are defective in the brain of patients with psychosis. And so this is a mouse brain, which basically it's nice because it's colored in a different way, and it serves the purpose of me telling you the relationship between striatal, that is in mice striatal, or caudal in humans, between striatal and cortical dopamine in the manifestation of schizophrenia phenotype. And it's been thought for many, many years, and I should say that nowadays, I was just at a meeting on schizophrenia last week, and there's a lot of very good evidence now that in schizophrenia, this condition that we coined as schizophrenia, there is too much dopamine tone in this area of the brain, whereas there is not enough dopamine here in this other area of the brain that we call prefrontal cortex. So we know, for example, that in schizophrenia, there are various phenotypes. The symptoms are different. We have positive symptoms that are hallucination, agitation, disorganized thoughts. These positive symptoms are thought to be due to the fact that there's too much dopamine, and I'll come back in a second on this. Whereas the negative symptoms, the reduced social and emotional expression, the lack of motivation, and many other deficits in schizophrenia are more akin to what happens in the frontal cortex, that is the low function of dopamine. So what I'm going to show you in the next slide is that we think now that schizophrenia is probably a circuit dysfunction. That is a circuit that goes from the cortex to the striatum, back to the dopamine synthesizing cells, back to the thalamus, and back to the cortex. And I'll show you that in a schematic way that there's a whole circuit here that is probably defective. And the way, and as you remember, many years ago when the genome was sequenced, people were saying, oh, we will finally identify the gene that causes schizophrenia. Now we understand there are many mutations that can lead to manifestation of the symptoms of schizophrenia. So and what we need to do is to find ways, because it's in practice drugs for 100 things like that, but there's still room to try to find ways for compounds that will alleviate in a selective and effective fashion the symptoms of schizophrenia. And you know, for example, that positive symptoms modulated by current do not do very well for the negative symptoms. That is, they hardly correct these symptoms. So if we had things that could manage the negative symptoms, would be a great improvement. Now, back to our ways of how we identified mice that are, quote, have psychosis. Now, we treat them with amphetamine, which is a psychostimulant, or which releases a lot of dopamine, particularly in this area of the brain, or we treat them with PCP. PCP phencyclidine is an inhibitor of the glutamate. Glutamate is the fast neurotransmitters in the brain, and glutamate is in this area, feeds back onto the striatum, and when we block the glutamate receptor with PCP, it basically eliminates the ability, or it recapitulates essentially the hypodopaminergic tone in the frontal cortex. And the reason why, the reason why this is valid is that I should say that all antipsychotics that are on the market have been developed by testing them in animal model initially against PCP and amphetamine. So amphetamine, when you give amphetamine or PCP exposure to individuals, even normal individuals, it can lead to schizophrenia-like symptoms. But it also, what it does, if people with schizophrenia might be exposed to amphetamine or PCP, it will exacerbate the symptoms in patients. So you see that it has some validity to using them in mice. Now, in animals, as I was mentioning a minute ago, we try to model the biochemical changes that we think are present in the brain of this condition. But we don't try to model the exact symptoms because it's almost impossible to do this. Although some of the social behavior or the motivation, and it can be modeled to some extent, as I will show you in a few minutes. So this is another rendition, shown in a diagram fashion of this circuit that I was telling you about, of cerebral cortex feeding to the striatum, back to the substantia nigra, and back to the thalamus, and this controls many behavior. The interesting thing is that this relay here of the striatum or the caudate has two different types of cells that express the D2 dopamine receptors that are in red here, and cells that express the D1 dopamine receptors. They're both stimulated by dopamine, but they are connected to the pathway in a different way. So you can see that the D1 are connected in the direct pathway because they feed back to substantia nigra, the other part of the brain, back to the thalamus, quite directly to the cortex. Whereas the cells that express the D2 receptors will project to this area of the brain, globus pallidus, back to the subthalamic nucleus, back to the substantia nigra, and back to the thalamus. So this is called the indirect pathway. And this is what antipsychotics do. They block D2 receptors and block mainly this pathway. So how do we assess these things in animals? How do we make sense of this? Because you can see that D2 receptors here in a schematic fashion, which this depicts this pathway here. There are D2 receptors on cells that synthesize dopamine that act to regulate the synthesis of dopamine, the firing of the cells, that is the release. There are cells on these striatal cells. There are also D1 receptors here on the striatal cells, but in the striatum there are other cells that use acetylcholine as the neurotransmitters, and they have dopamine D2 receptors, and there are also dopamine receptors in the cortex. So when you give someone an antipsychotic, you inhibit all of these receptors. So that the challenge for us to try to do this, to study this, is that we need animals in which we can remove the arrestin molecules or the kinase or some of these, or the receptors for that fashion, in a selective fashion. So Nikhil Erst in the lab generated in the past several years a flox, what we call a flox animals, and this gives us the ability to delete the D2 receptors in individual neurons of the brain. I'm not going to go through all the genetic technique, but just take it as to the fact that we can do this quite successfully in animal models. So the hypothesis that we have been formulating is that since clinically antipsychotics all interact with D2 dopamine receptors, we would expect that deleting the arrestin in the indirect pathway, that is this pathway that's downstream of the dopamine D2 receptors here in the striatum, would mimic the actions of antipsychotic. And so I'm going to show you here just very schematically I chose amphetamine just to show you how what animals do when you basically inject them with amphetamine. And here we calculate the distance that they travel when they're in an environment, in a square environment. And so when you inject amphetamine at this point, you can see that normally you put them in this environment, they will be a little active, but then they habituate to the environment. And then, when you stimulate with amphetamine, you can see that they have a really sharp increase in locomotor activity. They just run around like crazy. Now, here is one of the typical antipsychotic, haloperidol, Haldol, for many of you will recognize. When you give this at the same time as the amphetamine, you can see that you get a much, much decreased locomotor activity. So this is how we test the effectiveness of antipsychotic. Now, look at what happened when we knock out the arrestin molecule in these cells of the striatum. Well, it pretty much recapitulates what the haloperidol does, what an antipsychotic does. So we take this as proof that this pathway may be well involved in the actions of dopamine, in controlling locomotion, but also that eliminating the arrestin really recapitulate what antipsychotics do. And we can do this for phencyclidine. So what I showed you a few minutes ago in the other slides, we basically showed that deletion of arrestin or Barrestin in D2 dopamine neurons recapitulates the inhibition of the D2 receptors with classical antipsychotic in those pharmacological model of schizophrenia. So this is how we basically have shown that probably the arrestin is involved in this pathway that we had identified in modulating the effects, the physiological effects of dopamine, but also mimicking antipsychotic action. Now, what I'm going to spend the rest of my presentation is telling you about some of the efforts that we have made in trying to leverage this concept of functional selectivity, not so much to understand the cell and molecular basis of disease, which we're interested in, which we will do eventually, but we have focused the data that we have on the development of more effective and selective and effective therapies for hopefully treating schizophrenia. So we were kind of blessed several years ago, almost 10 years ago. The NIMH basically gave us a very large program project grant, which is carried out in collaboration with our colleague, Dr. Brian Roth at UNC, Chapel Hill, and Jian Jin, who was also at UNC, who is now at Mount Sinai. Jian Jin is a chemist, and also in collaboration with Dr. Bill Wetzel, who runs the behavioral core for mice here at Duke University, and ourselves. And initially, we were paired with Wyeth, the drug company, and Wyeth was eventually bought by Pfizer. And so we have been paired with Pfizer for this effort. And rest assured that Pfizer does not get any money from the government. What they do is they try to help us out to see how we could develop compounds that have potentials to be drugs, okay? So initially what Jian Jin did is that he took, and we had decided before we went to, we were working with Pfizer, we took the scaffold of aripiprazole. Many of you will recognize that this name aripiprazole is, the commercial name is Abilify, so this is the latest generation of antipsychotic. And why we took this is that aripiprazole seems to have a little bit of ability not only to block the dopamine receptors, but to activate the dopamine receptors. And we really thought that at this point, interesting to have this. And as you see, we've published this back in 2011. We have characterized more fully two different compounds, 9975, 994, which I will compare you in a few minutes. But in the process of developing this, we developed them. We didn't want them to activate so much the dopamine receptors, the G protein arm of the receptors. You remember, this is the canonical arm. We wanted them to be able to potentially activate a little bit the D2 arrestin arm of this pathway that we had discovered back in 2005, 2007, and things like this. And so these are cell assays that you see. Dopamine is very nicely able to stimulate the signaling molecule downstream of the G protein, downstream of D2 receptors. So is aripiprazole. It's a pretty effective, called partial agonist. It gives a partial response. But these compounds didn't seem to be very potent, which was okay with us. But in an assay which was quite spruced up, as we say, I mean, it was an assay to try to find, you know, the best in those compounds, how they could activate the pathway downstream of the D2 and the arrestin pathway. We were able to show that 994 and 9975 were partial agonists. They could activate this pathway. So we then basically, it behooved us to basically say, well, are these compounds of interest? Do they act as antipsychotic in animals? So again, we use amphetamine-mediated locomotion, as shown here. When you inject amphetamine, the mice run around like crazy. But then when you, at the same time, inject the compounds, you can see 9975 here, you markedly reduce the ability of amphetamine to elicit hyperlocomotion in those mice, which is a hallmark of antipsychotic. Similarly, we tested 994. I'm not showing you all of the tests that we've done, but we tested them in PCP-mediated hyperlocomotion. PCP, which I mentioned, is an inhibitor of the glutamate receptors of the fast neurotransmission, mostly in the striatum. You can see that it induces a hyperactivity, but not to the extent of amphetamine. But yet, when we put the 994 compound, it acts like an antipsychotic. It reduces this. But the interesting thing here is that when we do this in mice that are lacking the arrestin, which is the obligate protein for this pathway, you can see that now the compound is not active at all. So this really was a validation that these compounds probably do function as antipsychotic. And I'm not going to go through this, but these compounds seem to have less side effects. That is catalepsy, which is a hallmark of Haldol, for example. These compounds seem to have much less catalepsy, which was kind of interesting. So we showed that the D2 arrestin bias compound that we had developed behaved like antipsychotic in the amphetamine and PCP treated mice. But we also tested them in mice that I just mentioned a few minutes ago in the dopamine transporter knockout that we had made more than 20 years ago, which are very hyperactive because they have a lot of dopamine. But we tested them also in other mice that we had made that Amy Moan, or Amy Ramsey, who was a postdoc in my lab, had generated when she was a graduate student at UNC, and then she joined our lab and we characterized these mice. So these mimic essentially what you get with PCP, because this NR1 gene is a component of the glutamate receptors. So these mice have dysfunctional glutamate receptors. And look at what happened in these mice. They have a little bit of hyperlocomotion, just like PCP treated mice. But what they have is that this is wild type mice, you can see. And mice like to huddle together. I mean, you see mice, they're all going to be in the corner. Look at these mice that are lacking the glutamate output from the striatum. We call them asocial because they don't like to associate with each other. If you look, I mean, in the paper that we published back in 1999, we had a whole series of these mice, of these pictures, and none of them like to associate with each other. So, which is basically recapitulate that this is like a lack of social skills, essentially, in mice, if you wish. So the critical question here that we have after these data that I presented to you is, since the activity of the D2-arrestin bias compounds, okay, depend on the receptors being phosphorylated, being acted upon by these kinases, or that the arrestin was binding to the receptors to engage some of these signaling molecules. Will they have the same profile in every neuronal population? So this is interesting because I already told you that there's some really different findings in schizophrenia concerning the dopamine system. In one, there's too much dopamine in the striatum and too little in the frontal cortex. So how do we do this? How do we assess this? Well, basically we find ways to delete arrestin, for example, in specific neurons of the brain. And I mentioned this, that we now have a mouse that can do this. So what using this flox mice that Nikhil Hurst prepared or engineered in our lab, we then assess the effect of the bias ligands that we have developed with our colleagues at UNC, and also typical antipsychotic on response to, I'll show you response to amphetamine initially, but also we've done it in PCP, and I'll show you that in the next slide. So if you delete the Barrestin in all D1 neurons, there is no difference here. So the response to amphetamine, okay, and you can see that this is a concentration of the 3 mg per kg. They don't have any difference at 3 mg per kg. But haloperidol, aripiprazole, or clozapine can inhibit this response just as much in wild-type mice or in mice that are lacking the arrestin in D1 neuron. Now, the same thing is true if Barrestin is lacking in the D2 striatal dopamine neurons, or all the dopamine neurons in the brain that I showed you in that schematic, presynaptic, frontal cortex, and all the areas of the brain. All these antipsychotic, all are capable of inhibiting the hyperactivity induced by amphetamine. Now, we were quite surprised in this originally when we tested these two compounds, UNC 9975 and 994. 9975 still works just about like the antipsychotics, okay, the typical antipsychotic. But look at what happened to 994. We lose the ability of 994 to inhibit the amphetamine response, and this is shown over here, when it's deleted in the striatal neuron or in all D2 containing neurons in the brain. So basically, from this, we concluded that the striatal Barrestin is required for the actions of 994 on amphetamine response, okay? So that whether the receptors, the arrestin is expressed in the striatal D2 neurons, on all D2 neurons, it is required for inhibiting amphetamine. Now, we then did the same thing, but now on PCP responses. And here, we're not showing, I'm not showing all 3 antipsychotic, but I'm basically showing you that aripiprazole is effective at inhibiting the locomotor response that's induced by injection of systemic injection of PCP. And we already said that PCP in this type of model basically inhibits the glutamate receptors in the frontal cortex. But so what happened is that the 9975 has no difference. It inhibits not totally, but partially. But 994, when you look at 994 selectively, in the D1, if you delete it in D1 containing neurons, there's no difference. But then if you delete it in all the cells that are lacking D2, you can see that you lose the responsiveness. Whereas if you delete the arrestin only in the D2 cell of the striatum, it's still potent. So this was a little bit complicated. So we said, well, we want to test it if we only delete the arrestin in the frontal cortex specifically, or in the striatum. And I'm going to save you the idea because this is a lot of genetic manipulation. And what we did essentially is that we tested the effect of 994 to inhibit the PCP response when arrestin is removed only in the cortex, and you can see it's still there. But then when we remove it in the cortex and in the striatum, you can see now you lose the effect. So conclusions from these suggestive studies basically suggest that the cortical Barrestin for the UNC compound to inhibit the. Now, what could be the molecular basis for this? Because this is kind of shocking that you bind to receptors, and in one place you basically have a function, and then in the other place you don't. I mean, so it behooves us to try to find what the molecular basis of these different profiles are. So we went back to our characterization of the compounds in cells as we had done originally, showing that they didn't have much activity and that they were able to engage the D2 Barrestin pathway. And then we tested aripiprazole, which is the classical antipsychotic, and aripiprazole can inhibit the G protein signaling and also inhibit the D2 receptor interaction. The UNC compound basically is capable of inhibiting both, as shown here. And I didn't mention this, but we thought that these compounds initially, when we developed them, had very much the same profiles, the 994 and the 9975. And that's why we continued to use them. But then look at what happened with the 994 compound. It's capable of inhibiting the interaction of dopamine receptor with the arrestin, but it's essentially not capable of inhibiting the G protein signaling of the receptors. So we said, well, maybe this is modulated by the kinase. So do this pathway on this kinase. 9974 is not affected by the kinase, and 994, its ability or its inability to inhibit the cyclic EMP response or the G protein response is not modified, but its ability to inhibit the arrestin interaction with the D2 receptors is diminished. So in neurology, this is interesting because this diminishing an agonist activity means that you probably have the induction of a agonist activity, something that can activate the receptors. And so we concluded from this that UNC 994 behaves as an arrestin selective ligand with little or no efficacy at the D2 G protein signaling. So just to recap here, I mentioned before that this pathway really requires the modification of receptors with the kinase, binding to the arrestin so it can scaffold these signaling molecules. So therefore, we think that the levels of GRK and arrestin in a tissue should enhance the ability of a ligand to activate non-conventional signaling, that is that pathway here that we've characterized and is presumably sensitive to antipsychotic. Now, we went back, and I'm going to go rapidly through this, but we went back in our mice and showed that this is in the striatum, or the caudate, if you wish, in humans, that there's much less GRK2, this kinase that phosphorylates the receptors, than there is in the cortex. So there's more in the cortex. Same thing is true for Barrestin 2, which is the arrestin that's interacting with the D2 receptors. But it's not, it's really similar with Barrestin 1. Barrestin 1 is not different. Other people had shown that GRK2 are low in the striatum, but much more in the cortex, as shown here. We then went to look, we obtained from some colleagues human samples of prefrontal cortex and caudate, and we could show that there's much less kinase in the caudate than there is in the striatum, and there's much less beta-arrestin 2 than there is in the cortex of beta-arrestin 2, while Barrestin 1 is very equally expressed. So quite interestingly, it's expressed 3 to 5-fold more here than it is in the cortex than it is in the caudate in humans, which is kind of interesting. And not to belabor the point, is that when we test in cells, like in cells that we use in the lab, the ability of these compounds in the absence of a kinase to stimulate the interaction of this dopamine D2 receptor with arrestin, you can see that they're not active, whereas dopamine is very active. But then when we overexpress the kinase, you see that now we can reveal some activity of these compounds, suggesting that maybe this is what is happening. Maybe that this ability of the cortex to express more kinase and more arrestin will give compounds the ability to function as agonists in this area of the brain. Now, recall again this thing. So if this is dependent, this pathway is dependent on GRK, what about if you inhibit a GRK with a compound? There's a new compound that came out on the market that has a high affinity compound and selectivity for GRK2. And then, as shown in the dark line over here, when you stimulate the D2 Barrestin interaction with dopamine and you then expose it to the GRK inhibitor, you can see that it's much, much less active. Even if you overexpress with the, you shift the curves to more potent efficacy for dopamine, and you shift it to the right with the inhibitor of the kinase. Now look at what happened. We tested 994 in this paradigm. 994 alone doesn't do anything to the D2 receptor arrestin interaction, but if you overexpress the kinase, you can see that now you have a, quote, partial agonist effect, and it can be reduced with this kinase inhibitors, validating the point that the interaction of this compound here with the D2 receptor is highly dependent not only on arrestin but also on GRK2. Then we wanted to know, well, what about in what is happening in the prefrontal cortex? I mean, could UNC inhibit PCP response in the prefrontal cortex? And what is the effect, or is it dependent on kinase? So it had been shown in the literature that if you inject specifically in the cortex by doing bilateral injection in this specific area of the brain, of a mouse brain, you can see that you can reduce the ability of PCP here to enhance locomotion. And you can, so quinpirole is a full agonist at the D2 receptors, and you see it's effective at reducing the response here. Now, we did the same thing. We added quinpirole to our mice here, and quinpirole can inhibit, recapitulating what is shown here. And then look what happened with 994. It is capable of inhibiting the response to PCP almost as good as the full agonist quinpirole. Aripiprazole is a little bit less active. Now we said, well, since we demonstrated in cells that this is dependent on the kinase, we tested whether the inhibitor of the kinase was capable of reversing this effect of 994, and indeed it was able to recapitulate, to reverse this ability of 994 to induce a decrease response. Now, I'm not going to go into the detail, but we've also been able to delete D2 receptors, the receptor itself, either in the striatum or in the cortex, and measure the PCP response in these animals. And you can see that when you delete the D2 receptors, you get a marked reduction in the effects of PCP in the striatal, whereas you get a marked increase in the effects of PCP if you do it in the cortex, if you delete the D2 dopamine receptors. Just again, the idea that in the cortex and striatum, D2 dopamine receptors probably do very different things. So now, since I've basically showed you that there's a lot of things happening in the cortex, so we basically asked the question, well, what neurons in the cortex could be a or the possible site of action for these compounds? And we know already from classical studies that in schizophrenia, the markers for a cell type in the cortex that is called fast-spiking GABAergic interneurons. That is GAD67, which is the enzyme that synthesizes GABA, or ParvAlbumin, which is also a marker. They're reduced. These markers are reduced. That is, the cells are reduced. There's a reduction of these cells in people with schizophrenia. And we also know that these cells drive the gamma rhythm that people have shown that are disrupted in schizophrenia. So those are good models for potentially trying to see what happens with our tool compounds to these cells in the striatum, because we can show that these cells that are labeled with ParvAlbumin also express the kinase GRK2, and they express, you see the cortex and then the striatum. There's much more kinase expressed in the cortex. So what we only have a couple of slides to go. So what we did at this point is we turned to our colleague, Dr. Patricia O'Donnell, who used to be at the University of Maryland. And Patricia has studied for a number of years the function of these fast-spiking interneurons in the cortex with respect to trying to model schizophrenia phenotype, and also the other main neurons in the brain, the pyramidal cells. And Patricia had shown that when you activate the dopamine D2 receptors in the cortex, you activate these fast-spiking interneurons, you activate the D2 receptors with a compound like quinpirole, you can enhance the firing of these cells, and that's blocked with a selective D2 antagonist. On the other hand, pyramidal neurons, the primary cells in the cortex, are inhibited by activating D2 receptors. So you see they play different things. So what we did is that we convinced Steve Gee, who's a postdoc in Patricia's lab, to use electrophysiological experiment with using mouse cortical brain slices, okay? And the way that Steve can identify these cells, these particular cells, is that we have mice that are expressing one of the marker, the GAD67, so they are colored, okay? So he can identify and basically record from these cells, activation of these cells. And what you can see is that when he tested aripiprazole, which is the third generation antipsychotic, there's a little bit of an effect here. It stimulates the firing of these cells. And this is blocked by the same antagonist that I showed in the previous slide that is selective for blocking D2 receptors. Now, what happens in these fast-spiking interneurons with 994? Well, you can see here that 994, in comparison to aripiprazole, is much, much more effective as a stimulator. It enhances the firing of these cells quite a bit more than, say, the control or even aripiprazole. And if the mechanism is what I just told you in the last several slides, if the mechanism is through an arrestin and a kinase, you would expect that in mice that are lacking the arrestin in the frontal cortex, that 994 would have no ability to stimulate the cells, and indeed, it loses its ability to stimulate the cells, or it loses its ability if you inject the compound, like I told you a few minutes ago, compound 101, which inhibits the kinase. You see that you can inhibit this thing. So Steve then asked, in the striatum, does 994 do the same thing, or is it kind of more of an antagonist, which we think it is? So electrophysiologically, he was able to show that quinpirole is capable of acting as an agonist in the striatum. It basically inhibits the firing of these neurons. That's been known for a long time. But 994 is really not effective, suggesting that it's not capable of acting as an agonist in the striatum. As a matter of fact, it can actually inhibit the effects of quinpirole. Now, I think this is probably all the slides that I have to show you, the slides of data, and I'm kind of sorry that it was so heavy on data, but I think what I have demonstrated for you, I think we have accumulated evidence in the work that I presented to you, that tool compound that we call UNC994A basically functions as an antagonist in the cortex, but probably functions as an agonist in the striatum. So a compound like this probably would, if you have a functionally selective D2 ligand with agonist property in the frontal cortex, but antagonist in the striatum, we think that it would be an interesting compound to capture the complexity of the CNS with respect to the dopamine system in schizophrenia. So probably what we would expect that this compound like 994 should be able to potentially correct some of the social and emotional or motivation deficits or the cognitive deficits that are associated with the low dopamine in the cortex, because normal antipsychotic basically inhibit dopamine receptors everywhere. So basically they inhibit dopamine receptors when you should activate them in the cortex, and but they basically should inhibit the hyperdopaminergic tone. That's what they do best, but they don't take care very much of the negative symptoms of schizophrenia. So in the data that I've shown you, and it's, I hope you forgive me as this was sort of long-winded, is that functionally selective dopamine signaling pathways regulate specific behavioral outcomes, at least in mice, we know that. And that the UNC994 really provides a proof of concept that the D2-arrestin bias ligand can exert region-specific actions in the brain. That the Barrestin and the GRK2, those two molecules that I told you are so important in this alternative signaling of GPCRs, the levels in various areas of the brain could alter the drug response, like I showed you in the cortex versus striatum. And hopefully, we think that the D2 bias 994 may provide a path to the development of therapies with better specificity and efficacy in the future. So I would like with this slide to close my presentation and basically tell you that Nikhil Hers in the lab has carried the lion part of this project. Sean Peterson has also contributed to this. Sean had made mutant receptors that are themselves functionally selective. I didn't talk about this. Josh Snyder also was involved very early in the project, and several other people have contributed, and my long-term colleague, Dr. Barrick has also contributed to this. We could not have done this without our colleagues at UNC, Brian Roth, Jan Jin, who's now at Mount Sinai, and two fantastic postdocs that initially did the first paper, John Allen and John McCorvey, who's been instrumental in carrying these the last data that I talked about, and then colleagues at Pfizer, which have been very generous with their help and advice in trying to do some of these studies, and people that are supplied reagents, like Dr. Emiliana Borelli at UCI, who gave us the D2 flux mice. And I should not finish my talk by or I should basically acknowledge the work of Ramona Rodriguez and Bill Wetzel here at Duke, who are doing the behavioral facility for mice, and we count on them a lot all the time. But I should basically mention that this work has been supported in large part by a merit award that was given to our lab. Also, the award, the U19 project, which is done in collaboration with Pfizer and UNC. We also have a Center of Excellence from NIDA. I had I've been very fortunate to have an endowment from the PAL Family Fund. The Michael J. Fox has helped us tremendously also. And then I should say, as Dr. Barrestin mentioned at the beginning, we've had support over the years, not only for myself, as a grant for established investigator and award, but also many of my postdocs in the lab over the years have benefited from the money that was given in some of the grants for that many of you contribute to the success of the Brain and Behavior Research Foundation. And I'm eternally recognizing for their support. And I think I will leave you with this slide, which is my summary slide, and then we can basically finish with this. Okay, Jeff, if you want to take it.
Yes, thank you, Marc, for an outstanding presentation. I think that the key take-home message for a lot of our audience, I think, is that your presentation shows how very basic research can lead to the development of potentially new therapies, including medication, and that by understanding some of the basic aspects that you and collaborators have found, you could then move towards, based on that information, developing new treatment. And I think your last slide also shows how this is an effort that takes a team, and it's not one person sitting alone in the lab, but it is you and many others collaborating to develop this. And one of the questions, and we only really have time for one question, and the question I want to ask you is one that a number of people asked, which is, what do you see as the potential time frame to develop a new medication based upon these findings?
Well, I would say this is interesting. I mean, I don't know. I mean, I told you at the beginning what we were teamed up with the pharmaceutical company to carry this U19 project, and what the company does, and I made sure that I made the point that the company doesn't get any money from NIH, but what they do is that if there is what we call in science traction for the basic science that we do for developing, something that could eventually go to the clinic, is that they commit that they will sort of carry the project, in like phase 1 and the initial phase of trying to see whether there is value to developing these compounds. And that's very exciting because, as you know, Jeff, I mean, in academia, we don't have the whereabouts to do this. And many times, if we want to do these things, people have to start companies, and then we get out of our expertise, our level of comfort, So this is very exciting. And as far as and I cannot tell you whether things would be successful or not. I mean, I know that our colleagues at Pfizer are very excited about the idea, but it may still take several years, but it's worth waiting for, because this is in one of my slides I had the word, and I forgot to mention it. I had the name of Arvid Carlsson, and you know who Arvid Carlsson is. Arvid is a very, very good friend of mine, and you remember that he was the one that discovered dopamine back in 1958, and for that he got the Nobel Prize back in 2000 or something with several of his colleagues. And Arvid had basically promoted the idea back 20 that what we needed for to make a difference in schizophrenia, he called this, we need a dopamine stabilizer, because he had realized that that there was imbalance in the dopamine system in that give you the schizophrenia phenotype. And I think this is I'm very happy and very satisfied for him that if this approach that we have worked out may eventually work, because it would be something really very gratifying for the fortitude that he had to suggest these things. I'm not sure that what he was suggesting was exactly what we found, but there was basis to his fundamental understanding of the system. And I've been a student of his for a long time. Let's put it that way.
Well, I agree with you, and I think it's exciting to think of the possibility of new medication that potentially can have an impact on the negative symptoms, and you described them during the presentation, which often are the symptoms that are the most difficult to treat and are most debilitating. So it's something for us to look forward to and be hopeful about. So Marc, I want to thank you for your presentation and just as importantly for the extraordinary work that you and your team have done. I also want to thank our audience for joining us today. All of the research we fund is made possible through private donations. So if you'd like to make a gift, please visit our website, bbrfoundation.org, or call us at 1-800-829-8289. I hope you'll join us again next month when Dr. James Leckman, a member of our scientific council, a 1993 NARSAD grantee, and the Neeson Harris Professor of Child Psychiatry, Psychology, and Pediatrics at Yale, will present a webinar entitled Primary Prevention in Child Psychiatry, the Transformative Power of Children and Families. This will take place on Tuesday, May 10th at 2 p.m. Eastern Time. Once again, thank you, Dr. Caron, and thank you, audience. Have a wonderful rest of the day. Take care.
Thank you.